Hi everyone. Let's see if we can get this turned around. Let's see if we can get this zoomed in. All right. Literally, evening. Welcome to the lab exam study guide for camp, the camp test, the MSA mannitol salt test, and finally anybody, but you're more than welcome to share it. Literally just filming this on my phone, on a table, and on my computer. So, like, take down the main things that we and give this to you and try and do it in less than an hour. But at the end of the day, I like to be more precise, and then instead of just like trying to put it out there, I can at least be as concise as humanly possible. So this is the lab exam study guide, and I'm going to be covering Streptococcus, Staphylococcus. We'll be talking about bile escalin test, and then we'll be talking about hemolysis, which is the this is the camp test. So I'm going to zoom in so that you can see better. But if it ever gets like kind of tough to see, I'll try and like back up because I'm doing this from above. So yeah, we should be good. So let's start really quick with, and I'll even try to zoom. Okay, that should be good. What I'll do too is bring this up a bit so that it's better to see. Because what's the point of shooting a video if you can't see anything, right? So yeah. Okay, so starting with Staphylococcus, the Staphylococcus characteristics are composed of four main items. One is the fact that they are gram positive. They are also catalase positive. They exist on skin and mucous membranes, and finally, they are salt tolerant. So I made a really, really stupid way to memorize all this. And you might not honestly need all this, but sometimes if you want to memorize something, um, you just like can make a funny picture or a funny story, and it helps you memorize. So I'm gonna zoom in here on my little stupid photo. So essentially, I made Staphylococcus, Mr. Staphylococcus, right? Mr. Staphylococcus, and by the way, Staphylococcus aureus, the root word or is means gold. That's my understanding, and so you know this is essentially Mr. Gold, who this is his a hotel that he's staying at, okay? And Mr. Staphylococcus is a positive guy, as you can see his little positive sign, and you can see his little clusters. And he lives in the Skin and Mucus Hotel because that's where he resides. And so there's like coccus, but we'll get to that in a little bit. But essentially, um, the pathogenic side of Mr. Staphylococcus is aureus, and then the non pathogenic side is this staph epididymis, or SAFE, and this is non pathogenic. And what else did I say? So Staphylococcus aureus has a couple cousins by the name of, sorry, Staphylococcus aureus causes MRSA, septicemia, and toxic shock. Okay, and this is his he loves because it's high in salt and there's lots of bubbles. And so this is a synonym or analogy for something that's high salt and something that's high salt and also that's catalase positive. Because as I said before, number four here is that there's a high Staphylococcus aureus has a high salt tolerance. And it resides on the skin and mucous membrane. It's catalyst positive and it's gram positive. So I gave you the two versions of the pathogenic and the non-pathogenic. The pathogenic is aureus, and the non-pathogenic is Staphylococcus epididymis, which just resides on the skin. So they both reside on the skin. Facultative anaerobe is the word that's used to say that it's got a high salt tolerance, right? So here's another way to summarize that whole thing. Okay, so this might be a lot for you, or this might be not enough. It might be too, silly for you, but you know, if this helps, pathogenic bacteria who is clustered away that he loves. Cousin Staph epididymis is safe and non-pathogenic and grows in 10% NaCl, a yellow halo colony, and it's also positive in coag and positive DNase. This last part doesn't really make sense. All I'm trying to say here and remember is the fact that that it will show up as a positive test. So I didn't really find a creative way to put that in here, but the main things that you need to know, the main characteristics that you need to know about Staphylococcus aureus is that it's gram positive catalase and there's high salt tolerance. So okay, so here's the Staphylococcus species. Excuse me, Streptococcus species. We know from the last video or one of the intro videos the fact that Staphylococcus resides actually is kind of, it's not that it's opposite of Staphylococcus, but there's just a lot of similarities, and then there's a lot of like direct kind of opposites. Like, for example, Streptococcus is gram positive, just as so it does not thrive in salt, whereas Streptococcus. And then you have its home. So that's kind of like more or less easy to remember these two. And sometimes, like when you make association, any kind of association with body parts like sex or something scary, it's a lot more likely for you to remember that. But if you have to put down a location of where these bacteria reside, those are the locations that we've read and that they've been found. So again, they exist in pairs and they also exist in chains. And that's my pair. I drew them myself, so I'm super proud. And here's a little summary. Strep is a positively negative catalase, has salt. Here's the thing. So Streptococcus species are extremely virulent species, right? They cause a lot of damage. Okay, when it comes to strep, there's a couple diseases that are probably important to know that they cause. So we'll start pretty, outright, and then I'll show you a way that I remembered it using the same chain. And it might not make sense, but you can come up with your own analogies or, however you need to remember it. So, Streptococcus mutans causes cavities. So, can I show that again? Streptococcus mutans causes cavities, and Streptococcus pyogenes is, can be found in the pharynx. Streptococcus pneumoniae can cause meningitis, otitis media, and what is that? What is this? Sinusitis. I could not think of that word, even though the word is right in front of me. So if you look at the end of these words, I don't know, depending on how much healthcare experience you have, if you see anything that ends with itis, it's essentially means inflammation. So sinusitis, otitis media, meningitis, these are all forms of inflammation. Otitis has to do with ears or the ear canal, sinuses, meningitis. So these are all forms of inflammation. So we'll keep going on the list. Streptococcus agalactiae is responsible for neonatal sepsis, which is extremely scary. For mothers out there, even fathers, you would want your little one to be safe, and there are certain strains of bacteria out there that are really very just virulent on you know, so I hate talking about it, but it's something to acknowledge and know, and so that something can be done about it. And doctors have, and so have researchers. So, and now we know what agalactic, agalac, agalactate causes. So I'm just learning this. I'm a student as well, so please forgive me for some of my mistakes and tripping over my words. I'm still learning some of the pronunciations and how they fit into the real world. So thank you for your patience. Okay, continuing, we have the species. Excuse me. We have Streptococcus species, right? That's what we're talking about. So we have Streptococcus faecalis, which is also known as Enterococcus. So faecalis and Enterococcus is the most recent way it's pronounced to understand. And actually, Enterococcus can cause food poisoning, UTIs, and endocarditis. Okay, so here's what I made over here on the left. It's kind of like a little diagram, and it's a whole chain with the things that it can, with the name of the species and what it causes. So, like I have here, mutins is oops. And then we have Algalactiae, which is associated with neonatal sepsis. Faecalis enterococcus is associated with food poisoning. This is really gross. Endocarditis and UTIs. And yeah, like I said, like kind of a way to show that how this where these bacteria, it can reside in the vagina, and it can reside in the mouth. And so I have a kind of a little mermaid there to unfortunately be my model to show where these things are affecting people. Going further, we have Streptococcus pyogenes. Like I said, that it affects the pharynx, and then finally pneumonia. Streptococcus pneumoniae can cause meningitis, media, and sinusitis. So I have a couple, just a couple of ways, fun ways to memorize these, because this is really interesting stuff to me anyway. I'll try and speed through these last ones. The last that we have left is MSA, bile escalin test, and finally hemolysis. When it comes to MSA, we're talking about the mannitol salt agar, and I think there's more or less six know about mannitol salt agar. Okay, so I drew a little picture of mannitol salt agar, hoping to show what it looks like, what a positive test looks like, and we'll just go from the first thing. First thing that you need to know about MSA mannitol salt agar is that it has the carb mannitol. Mannitol is a type of sugar. Okay, and we have mannitol, which is the type of sugar, and the role of mannitol is, sorry, so let me start back. So mannitol salt agar has mannitol, which is sugar, salt, which is NaCl, and a pH indicator. The pH is phenol red. So what is the purpose of the salt? The purpose of the salt is that it makes it a selective agar. And why is it selective? So I have these two things like fighting, but I don't know. I just thought it was really funny because this is a salt shaker and this is sugar and it's mannitol and they're fighting. So anyway, my point is salt is selective, and the reason why NaCl is in MSA is because it's selective, and the reason why it's selective is because most bacteria cannot survive in high salt, and so the fact that organism can survive on this agar is the fact that it's selective because it allows for this whatchamacallit, excuse me, it allows for this organism to thrive. Just one second. Okay, we are back. All right, so moving right along. I'm going to talk to you next about mannitol. Why do we have mannitol in the agar? So we have mannitol. The point of mannitol in MSA is differential. So mannitol's point of being there is that it's differential, and it essentially provides a substrate for fermentation, right? And I'll get back to that if that doesn't make any sense. And we'll talk about the thrive. So number four, I essentially said that Staphylococcus aureus is able to survive on and thrive to on high salt areas, and I did say that it's able, and the reason why it's able to thrive is it's because it's able to adapt to high salt areas. Like for example, for our skin is actually incredibly salty, and a lot of like really pathogenic bacteria can't survive on there. But aureus and I also believe Staphylococcus epidermidis. Right, and so that's why I have a picture of a butt and the skin, and the Staphylococcus is on there. Just kind of a funny way for you to memorize that, like, look, there's a salty butt, and the Staphylococcus is able to survive and thrive there. So there you are. Um, like I said, I make up stupid stuff so that you can memorize. And if it's helpful, good, and if it's not, then you gotta make up your own. I'm just helping you with videos that might help me. So, you know how my brain works. So continuing, we didn't talk about the pH indicator. So, phenol red is a pH indicator in MSA, and it's essentially a positive result is yellow acid, and that basically means yellow acid mannitol is fermented. Yellow acid mannitol is fermented, and I say that three times because over and over and over again until it just becomes ABC, and it becomes kind of comfortable in a way so that you don't have to struggle later on a test. So yeah, if your growth on the plate, right, this one right here, if the growth is pink, that's pretty much indicates that it's basic and you will have no fermentation. So if it's pink, it's basic and no fermentation. Did we already cover five? Yes, so we talked about the indicator. And now I have a question for you. All right, and I'm going to hide the answer. Okay, you probably already saw it, but don't look. Try and think about the logic behind it. Okay. True or false? MSA is used for determination of mannitol formation. Let me read that again. MSA is used for determination of mannitol formation. Fermentation. Three, two, one. It's false. Why is it false? It's because MSA is used to differentiate aureus. MSA is used to differentiate aureus. Um, oh, excuse me. It's like 1:30 a.m. and I'm sharing this video. Hopefully y'all appreciate this, whoever is gonna check this out. But essentially that's like it, go back over those questions. You can make them into questions, try and make them make sense. They are pretty straightforward, but I think that's what's going to be important on here. All right, and I just have a couple pointers for bile. So now we're talking about the bile escalin test. Escalin is actually escalatin and glucose combined. And the whole point of escalatin is that escalin is actually tree bark. It's made from tree bark. And so escalatin and plus gluten plus glucose, escalatin plus glucose make escalin. So next we have, in this agar, we have bile. And the whole point of bile is that it separates group D strep. And ferric citrate is essentially is oxidized iron. So we'll keep going down this as we go along, just to understand why. Oops, just to understand why it is we have this test. Okay. Um, okay, we're back. And then we have the fact that hydrolysis occurs under acidic conditions. And so again, what's the point of this test? Hydrolysis occurs under acidic condition, which makes D-glucose and esculin, right? This whole hydrolysis phenomenon that occurs in the bile test, it makes glucose and esculin. So hopefully that's clear to see. And then the thing that's unique about bioesculin is that few organisms can hydrolyze esculin in bile, and it makes it selective and differential. And so if we have, for example, a pure culture, then we're going to utilize a slant. Whereas if we have a mixed culture task to isolate, we're going to actually so if we have a mixed culture, the whole goal is to isolate the mixed culture, and then we want to find Streptococcus and Enterococcus. So a positive isolation, is to isolate EU faecium and group D strep. This is the last one before we are done for the day, but I'll give you the meat and potatoes of this, and then we're here in the home stretch. So, hemolysis. And hemolysis is kind of breaking open blood cells. Hemo that you're talking about, the heme, the blood cells, and then lysis means to cut or to break open. So that's our auger right there. And essentially, let's talk about a positive result. A positive result for hemolysis test is that an arrowhead zone of clearing will appear. It'll essentially look like this arrow that I drew here. So an arrowhead zone of clearing will appear, and the camp. So we have the camp, right? algalactiae, and we have beta hemolysin, which is aureus. Those, the algalactiae and the aureus, will synergize together and create an arrow shape. I haven't been able to find any research as to why, but if anyone can share, I'd be totally appreciative. I'm up for not only just teaching on this channel, but for learning. So those two go hand in hand. So yeah. So again, we have the camp that's associated with the agalactiae and is positive with the beta hemolysin or the aureus, and they synergize to make this. Do you like this? By the way, this is a bow and arrow. So it they synergize, and the organism is able to make a clearing, and that clearing of an arrowhead is a positive result. Organism protein. Other species essentially do not. And so you have right here this example. There's a streak here of aureus that is down the middle, and there is a algalactiae which is right here on the left. And you have S. pyogenes. All three of these species, right? The aureus and the S. algalactiae create, as I've drawn down here, an arrowhead like view. And because of this fuse together and create, it's called a positive test. And I believe this is a differential. I could be wrong. Pretty sure this is a differential to pretty much highlight the fact that you have obtained the presumptive ID of a galactiae. So that's my understanding of it. And that literally my talk. If this was helpful, let me know if there's another format that you prefer. But I just want to say thank you. Feel free to subscribe. Feel free to check it out. I do this every so often because honestly, it helps me. If you have a chance, my advice to you for this quiz is to go out and do one thing. You've listened to this recording on lab that composed of camp tests, MSA, and finally streptococcus. We talked about bile as well, kind of very briefly. What I would do after this test is one, go teach someone what you learned. Number two, okay, after you teach someone what you've learned, make questions from the video. Three, quiz yourself. Not treat yourself, but quiz yourself. And the reason why you need to quiz yourself is because when you, or you could call it a use it or lose it curve. And this curve is exponential. I take credit for this use it or lose it curve. That's what me and actually a friend of mine dubbed it. We've dubbed it the use it or lose it curve. You could also call it the forgetting curve. I think the curve goes by many ways. But the reason all of these are essential is because when you're exposed to something the first time, it's new, it's genuine, it's cool, and then it's like goes right out of your memory. But the way to maintain, or at least to do this, you might not like just completely lose it, but the way to slow down this curve and kind of have it do that, and essentially eventually have it do this. If this line right here is representing your ability to retain the information. This right here, right, is just, this right here is essentially time that passes. And this right here is your ability to retain the information, right? So over time, it's quick to forget. But if you do things to try and make yourself remember the information that you just learned by one, going to teach someone, I don't care. Literally be teaching, go in front of mirror and teaching your own self. Number two, make questions from the video. If I gave you some information that didn't make sense, make a question about it and then quiz yourself on it and then make sure that's right. Number three, which I just wrote number and I didn't write three. You guys are bogus for not letting me know. Whatever. Quiz yourself. You made those questions, now quiz yourself. How well do you know the information that you just learned? How are you going to be able to retain the information that you just learned? This information that you just learned so that when you take the test, this is not like you're reaching, struggling to find the answer. The answer will be there right on the surface. Just if I ask you ABC, what's after D? You can right away tell me the letter that's after D. And if you can't, then, well, maybe there's some other things you gotta look into. But if this was helpful, go ahead, give me everyone to do well. So thank you. Please subscribe, as we say. I like to share the knowledge I've learned and try and make it accessible to everyone because everyone should be able to learn. So I hope this was helpful. Take care. I kept this under 30 minutes. If in future videos I am happy to do that. It is like almost let me show you what time it is. It is 1:30 in the morning. So if you guys don't think I don't love you, I mean, look at that time. I value my sleep. And I'm out here in these streets teaching. Okay, I'll talk to you soon. Feel free to review this. And even if you don't have the energy to review this and do this three things that I said, just listen to it again and just do, it's not active learning anymore. It's called passive learning if you're just listening to it. But the way you make it active is to actually go and carry those things out. So, okay, take care, guys, and we'll see you next vid.